So good morning, everybody, and welcome to the joint seminar of the survey and the Hebrew University, both institutes. Today we have Yael Leshno as our speaker, and Yael Edelman will present her. So go ahead.
Hi, I'm delighted to introduce Yael Leshno to give her doctoral seminar. I first met Yael as an enthusiastic student of an undergraduate course at the Marine Station in Eilat. After finishing her BA in geology at Ben-Gurion University, she approached me to start a master's project under the supervision of Professor Chaim Binyamini and myself as a collaboration between the Geological Survey and Ben-Gurion University. Her master's work involved the use of young fossil records to identify anthropogenic change in the East Mediterranean. Yael finished her master's with high distinction, and her thesis produced 2 publications. Yael continued for a PhD at the Hebrew University and the Geological Survey under the supervision of Professor Rivka Rabinovich and myself. Yael dove deep into the marine fossils of the Jurassic, combining fieldwork, museum collections, and computational geoscience in order to answer questions regarding local and regional depositional environments, and also track global paths of faunal dispersion. In all of these aspects, Yael was a motor of innovation and led her own unique path. During her PhD, Yael was awarded the EU Synthesis Project grant to travel and work at the British Museum to do research. She received the Meidan Fellowship for doctoral students and also the Levi Eshkol PhD scholarship. We are very pleased to see that our collaborators, some of them could join us today and hear Yael's talk. I'm happy to add that Yael is also 8 months pregnant and might experience false labor contractions during her talk, and will have to stop for a few moments. But don't worry, we will use that time to think of names for the baby or something like that. So let's hear from Yael about the paleoenvironmental reconstruction of the Middle Jurassic using benthic macrofaunal assemblages from outcrops in northern and southern Israel. Please.
So thank you everybody for joining us today. Thank you, Yael, for that warm introduction. So I'm happy to talk about my work on the Jurassic. So let's dive in. The Jurassic is considered a greenhouse warm period that encompasses faunal and geodynamical changes that mark the onset of the modern world. During that time, reefs formed broad bands along the southern and northern margin of the Tethys, as we can see here in the colorful dots on the paleogeographic map. And these sedimentary rocks were extensively studied on a global scale due to their high economic potential, as they comprise approximately 25% of the world's petroleum reserves. And you can see the distribution of petroleum reserves here in the black spots on the lower left. So the Jurassic of Israel was studied by many authors. During the Jurassic, Israel was located on the southeastern margin of the Tethys at the northwestern edge of Gondwana, close to the equator. And as I said, the Jurassic sequence of Israel was extensively studied due to its high economic potential, with research mostly focused on boreholes from on and offshore. And the overall Jurassic sequence was thought to represent a shallow marine platform that passes into a deeper basin to the west. However, despite this general facies model, there are still open questions regarding the depositional environment of this sequence, as there's no detailed information on how the depositional environments of these rocks vary spatially, both on a local and regional scale, specifically regarding the regional correlation to the economic Arabian carbonate system. So in addition to all this knowledge on the lithology of the Jurassic, The Jurassic sequence of Israel is rich in fossils of many taxonomic groups, including benthic macrofauna such as bivalves, gastropods, corals, and somnoporoids. And past work on the fossil assemblage focused mostly on the biostratigraphy of different taxonomic groups, including foraminifera, ostracods, ammonites, and nanofossils. However, for the Jurassic sequence of Israel, there's no quantitative fossil information as a means for paleoecological reconstructions, meaning no study has any quantitative information regarding any of the macrofaunal data. So ultimately, the quantitative high-resolution paleoenvironment of Israel, of the Jurassic of Israel, remained largely unknown. So in this talk, I will present how I developed and applied new geoinformatic tools that use fossil assemblages to reconstruct the paleoenvironment of the Middle Jurassic of Israel. I focused in my work on fossil elements that characterize the Mesozoic shallow marine environment, concentrating on the abundant fossils of several taxonomic groups, including corals, somnoporoid sponges, bivalves, and gastropods. So what can we actually learn from fossil assemblages? Why should we study them? Macrobenthic invertebrates are a major component of sedimentary rocks due to their high preservation potential. They include a wide range of species that can be found in very diverse habitats, and most importantly, they're known to be sensitive indicators of seafloor conditions, such as food availability, water energy, substrate, water depth, and substrate. And using multivariate analysis of community structure and composition of the fossil assemblage, we can reconstruct the environmental conditions of different time intervals. So this works to our advantage. And my research focused on two exceptional outcrops of the Middle Jurassic in Israel, in the south and in the north. The southern outcrop of Makhtesh Gadol. Makhtesh means an erosional crater. And Makhtesh Gadol is located in the northern Negev area of southern Israel. And it forms one of the most complete and exposed sequences of colluvian strata in the Middle East. The northern outcrop of Mount Hermon is an uplifted structure that exposes colluvian to Oxfordian rocks. And both of these outcrops are very rich in fossils that have not been studied quantitatively, and we can use these to reconstruct the paleo environment. So let's dig into each of the outcrops. So the Jurassic of Makhtesh Gadol was originally divided by Goldberg to the Zohar, Kidod, and the Beersheba formations. So from right to left, we can see the limestone and marl of the Zohar formation, the overlying shale Kidod formation, and finally on the left here, the marl and limestone of the Beersheba formation. And Goldberg divided the section to lithological subunits according to changes in lithology and correlation with electric log markers from boreholes. And we can see the columnar section of Goldberg here on the left. And I added here in the bottom representative examples of Jurassic corals from each of these formations in the Makhtesh. The northern outcrop of Mount Hermon exposes, as I mentioned, colluvian to Oxfordian rocks from J4 to J6 that are very rich in fossils. So from the right to the left, we can see the limestone Hermon formation, J4, that's overlain by the shale Kidod J5, and the limestone Beersheba formation J6. The northern outcrop, as you can see from these photos, is much more lithified, has a relatively uniform lithology, which makes its subdivision much more difficult. And the fossils are much less accessible in this outcrop, much more poorly preserved than the southern outcrop of Mount Hermon. And here we can see a facile coral, a stomatoporite sponge, and a bivalve that, from each of these formations. So my PhD was divided to several projects, several chapters, and today I will present two of those. First, I will talk about my work on the Middle Jurassic patchy reefs of Makhtesh Gadol. After that, I will discuss the use of virtual paleontology and geometric morphometrics to identify Norenikin Gastropons, and we'll talk about these later. And in the remaining time, I will briefly show my current work on using machine learning to classify images of Norenikin Gastropons, of these fossil snails, to test for climatic change in the Middle and Upper Jurassic. The fourth chapter that I will not present today concentrates on the biogeographic distribution of macrobenthic assemblages from the Jurassic Boreal and Tethyan realms. So let's start off with the first chapter on the patchy reefs of Makhtesh Gadol. And this work was conducted in collaboration with Mark Wilson from the College of Worcester, Ohio. And if Mark is here today with us, I want to thank him for waking up in the middle of the night. And I also saw Bernard was here today. So Bernard Letoulier from the University of Lorraine, also a collaborator on this project. Previous work on the macrobenthic fossils of Makhtesh Gadol focused mostly on descriptive and taxonomic observations of different taxonomic groups, including mollusks, stomatopoid sponges, and corals and associated hard-bottom fauna. However, there's no detailed quantitative information on any of the fossil groups, and most importantly, on the biogenic structures that they form. So the goal was to reconstruct the depositional environment in high resolution based on multivariate analysis of the fossil assemblage. And to do so, I sampled in high detail the fossiliferous horizons of Makhtesh Gadol, following Goldberg's lithological subunits that you can see here that are marked in blue, following the lateral exposure of the subunits in the Makhtesh, throughout the Makhtesh. So diving into the field work, I sampled the target patchy reefs using the standard accepted methods for studying fossil and modern reefs. I employed a stratified random sampling plan using quadrates, so 50 by 50 centimeter quadrates that you can see here in the photos, and using 10-meter line transects here on the right to quantitatively assess the diversity of the fossil assemblage. And from each quadrate, I identified all the faunal components, including the corals, the sponges, bivalves, and gastropons. And in addition to identifications, I photographed each quadrate, and then I used image analysis software to estimate the percent cover of coral and sponge components using a grid of 400 cells. So you can see this image from the software with the cell grid. Additionally, I used 10-meter line transects that were spaced in 10-meter intervals to estimate the diversity, abundance, and colony size of the corals and sponges in the patchy reefs. Specimens were identified to the lowest taxonomic level possible. And coral specimens, for corals, this is the genus level in the Jurassic. So coral specimens were identified from the weathered surfaces and from polished slabs. Here you can see me cutting a coral colony. And here on the bottom right, we can see the polished surface of the coral versus the hand sample and the field view of the coral. For the stomatopoid sponges, which are a group of Mesozoic carbonate sponges, they were identified based on their gross morphology and from polished slabs as well. Coral identification focused on the colony morphology, or the level of corallite integration and overall growth form, which are both used as a proxy for environmental conditions. So from these different structures, we can learn on water depth, salinity, nutrient levels, turbidity, and other environmental parameters in which the coral lived. For example, branching forms, branching corals, are more common in environments with a high sedimentation input. So diving in to the quantitative analysis of the fossil assemblage, we can look at the sequence in very high resolution. So on the left here, we have the columnar section with the studied subunits and the different colors. And from the quadrate analysis, we have the percent cover of coral and sponge components in each subunit. Remember the image analysis software. So here we have the coral cover in blue and the sponge cover in orange. And we can see that there's a shift from corals in the patchy reefs in the lower part of the section here to stromatoporides in the upper part of the sequence. And interestingly, there's this interbed of oolites that shows a shift back to corals in between these stomatoporide dominated units. From the quadrate analysis, we also get the species diversity. So focusing on coral diversity here, we can see that overall the species diversity in the sequence is equivalent to other reefs that are known from the southern Tethyan reef belt at that time. But we can see there are two subunits, two lithological intervals with very high coral diversity. And I'll address these in a minute. From the land transects data, we can see the distribution of the colony size in the section for the corals in the blue and the sponges in the orange. And the relatively constant colony size, both for corals and sponges throughout the sequence, indicates that we could rule out sedimentation as the limiting factor that's restricting the success of the coral and sponge growth. So these results show for the first time quantitative evidence of this shift from corals to sponges upsection. So to test for changes in the community structure and composition of the fossil fauna, I performed multivariate analysis. So this looks at not only which species are present, but what kind of grouping can we look at in the section. So here we see the results of the cluster analysis. And result of the cluster analysis is a dendrogram or a tree diagram. And cluster analysis basically groups samples based on their level of similarity in species composition. So the x-axis here represents the full set of samples, and the y-axis defines the level of similarity at which samples are grouped or clustered together. And the different colors of the samples correlate to their various locations within the section. So from the cluster analysis, we see that the samples group into natural 4 clusters or 4 groups, arranged by their location within the sequence. And these clusters are also statistically significant from each other. So starting off on the left here with the first cluster, I named it the coral-dominated A cluster. And these samples that constitute this cluster, they all originate from the subunits with the high coral diversity that we saw before. And these are represented in the columnar section in the dark blues. The second cluster includes samples from patchy reefs with the lower coral diversity in the lighter blue colors here. And next, we have the subunits with the high cover of sponges from the upper part of the section, and these all cluster together to the sponge-dominated cluster. Finally, all the samples from the oolite subunit form this distinct cluster with a distinctive coral composition that is unique to this lithological bed. Another way to visualize the multivariate data is through an MDS ordination or a non-metric multidimensional scaling ordination. And this visualizes the level of similarity between samples in a 2-dimensional map. So this gives us, unlike the cluster analysis, this gives us a continuous representation of the similarity. And in such a way, again, that the distance between pairs of samples reflects their relative similarity of species composition. And we can superimpose the clusters from the cluster analysis onto our ordination. So we can see here a gradient from coral-dominated to sponge-dominated subunits. And again, the oolite unit is all separated by itself. So for the first time, we see that the fauna divides the Machtesh Gadol section to statistically significant groups. And using a similarity percentage analysis or SIMPER analysis, we can calculate which taxa are primarily responsible for the observed difference between the groups of samples. So here we see the different genera that characterize each of these clusters. So coral cluster A is characterized by a group of corals called microsalinids. This is a unique group of deeper water corals. The branching corals, most dominantly by Lochmaismelia, characterize the second coral cluster, so coral cluster B. And obviously, the sponge-dominated cluster is characterized by the stomatoporeids, so monospecific aggregations of stomatoporeids from the upper part of the sequence. And finally, the Ula unit, as I mentioned, has this distinctive coral assemblage and is characterized primarily by the ceroid coral species Dendrolia. So if we switch now from looking at which species constitute each cluster to what are the morphologies that can be found in each cluster, we can see these two criteria in terms of colony structure, or as I said, the level of corallite integration within the coral colony and the overall growth form. And we can see that coral cluster A is dominated by massive forms in gray, with a very diverse and balanced distribution of the different colony structures. And in contrast, coral cluster B is dominated by branching forms in blues, with colony structures that are more adaptable to high sedimentation rates and turbid environments. And finally, the corals within the sponge-dominated and the Ulite unit clusters comprise of dendroid and ramos forms that are known from shallow and high energy environments. So now if we integrate all of this univariate and multivariate analysis of the fossil assemblage, we can reconstruct in high resolution the temporal variation in turbidity, nutrient availability, and water depth throughout the sequence. So if we start out here in the Zohel formation with the first patchy reef interval, that's associated to the coral cluster B in light blue. And that showed us lower coral species diversity, and it was comprised of branching forms with colony structures that were more adaptable to high sedimentation rates and turbid environments. And then in contrast, we have the next patchy reef in Goldberg's Kidot formation, and that belongs to coral cluster A in the dark blues. And that showed primarily massive domal microsalinids, and it had a very high species diversity and a variety of morphologies that suggests that these deeper mesotrophic conditions were more favorable for coral growth. And the next couple of patchy reefs shift back and forth between these two clusters. And the upper part of the sequence, as I mentioned, is composed of these thick monospecific aggregations of the branching stomatoporeids, with a low diversity of some ramos and dendroid corals that are tolerant to shallow, high energy environments. And in between the sponge units, we have the Ulite unit, and that is characterized by a distinct assemblage of corals from high energy environments. So we can compare the environmental reconstruction from the coral assemblage to data we have on the stable oxygen isotope record from the same lithological subunits. I collaborated with Matthias Alberti from Kiel University to reconstruct the sea surface temperature of the Jurassic of Israel from calcitic fossils. And the results of that study were published on the cover of Geology in last December. So overall, the temperatures for the sequence were high, as we would expect for the equatorial position at the time. If you recall, we're located pretty close to the equator. And interestingly, the coral cluster A, the one that had the lower diversity and more stressed environment indications, correlates with the highest recorded temperatures that reach as high as 35 degrees Celsius. So such high temperatures cause environmental stress and can probably account for the low diversity of the patchy reefs during this time interval. So if we switch from looking at the temporal to a lateral point of view, we can reconstruct the facies model for each of the clusters. And starting at coral-dominated cluster A, from the deeper shelf environments with the higher coral diversity to shallower, more high-energy stressed environments, and then shallowing upward in the section to an oolite shoal and lagoon environments that were rich in stomatopoid sponges. And this facies model is supported by the other faunal components, including the dominant mollusk groups in the section that also correlate well with this reconstruction of the depositional environment. And I just want to grab your attention here to the Norenids in the lower part of the graph, and we'll discuss this really high diversity of Norenids in the next chapter of the talk. So the shift from the corals to stomatopoid sponges in Israel and the species composition that we saw now can be highly correlated with a similar shift in the Colluvian Exfordian rocks of Saudi Arabia. So here we can see the Tuaik and Khanifa formations that are exposed in outcrop in this columnar section near Riyadh. And they show a similar shallowing upward trend that starts with corals and terminates with a high dominance of stomatoporides in hypersaline and oligotrophic lagoons. And these stomatoporides from the outcrop are known from about 300 kilometers west in the subsurface, here the columnar section here on the right. And these stomatoporides-rich units, notably of Cladocoopsis, are responsible for the high porosity in the subsurface of the Arab D reservoir, which is part of the Rawar field, one of the largest oil fields in the world. And you can see here in the thin section on the bottom, this shows the porosity in blue, and the porosity is primarily within the stomatoporide structures. So the high resolution reconstruction of Mahdi Gadol enables us a better correlation with the Arabian plate carbonate platform. So it's worth noting that this faunal change that is first described in Mahdi Gadol is related with such high primary porosity in the subsurface in Saudi Arabia. So to conclude this part, I've shown for the first time quantitative evidence for a shift from coral-dominated to stomatopoid sponge-dominated patchy reefs in the Israeli colluvian that indicates a shallowing upward sequence. And this high-resolution facies model correlates well with similar faunal shifts on the carbonate platform in Saudi Arabia. So moving to the next chapter, from my detailed analysis of the fossiliferous intervals of Mahdi Gadol, I saw that the most abundant fossils, numerically abundant fossils, were Norenikin Gastropons. So these fossil snails, superfamily Norenikidae, commonly known as Norenids, were one of the most diverse and abundant fossil groups of the Mesozoic. They were widely distributed across paleo latitudes, as you can see here in the paleogeographic map. All the blue dots represent locations of Norenids that were studied. And they're considered key faunal elements of Mesozoic carbonate ramps and platforms. And they were extensively studied and used for biostratigraphy and paleoecology. And here on the right, on the left, you can see photos of specimens that I collected from fieldwork in Mahdi Gadol. Just to illustrate how common these fossils are, you can see on the left here images of Norenids that I photographed from the flooring of the buildings in the university. This is from the Springsack building. So Norenids are distinctively recognized by the presence of these internal structures called folds that minimize the internal cavity of the shell. You can see these here on the cross section. Typically, specimens are cut or polished to reveal the internal fold morphology. And the number and the position of folds within the internal whorl outline form this essential component for the description and identification of species. They form the basis for their classification. However, the uniform classification of the group is limited due to its inconsistent systematic position and the fact that to identify these structures, you need years and years of experience, so expert experience, and the identification process is pretty difficult to replicate. So from my field work, I had to think of a way to identify these abundant fossils to use them for paleoecology. So my goal for the next chapter, for the second chapter, was to develop a new non-destructive method to quantitatively identify nerinids based on shape analysis using a geometric morphometrics approach. This project was in collaboration with John Todd from the Natural History Museum in London and Hilam Mai from Tel Aviv University. And I received funding for this project from the EU Synthesis Project. And this project was actually published in Paleontology, in the journal Paleontology. So I compiled a database of 58 specimens of nerinids, from which I extracted 157 internal whorl structures, internal whorl cross-sections. And these specimens cover a wide range of middle upper Jurassic Paleo latitudes in the red dots in order to capture a wide range of shell morphologies from the cross-Tethys shelf. And the specimens belong to 5 of the 7 known families of nerinids, which were also present in Israel. I used micro CT scanning at the Natural History Museum in London to scan fossils of the Natural History Museum fossil collection and both from fieldwork in Israel. And in addition to micro CT scans, I used photos of specimens of polished slabs from various sources, including specimens from paleontological collections, fieldwork in Israel, online databases that include photos of cross sections of specimens, and published plates in the literature. So now I'll walk you through the workflow for capturing the internal whorl outlines using this geometric morphometrics approach. So on the left here in A, we have the external view of the selected specimen, doesn't look like much. But after scanning, we have the 3D reconstruction from the CT stacks. And I show this here with a corner cut that shows the internal morphology of the gastropod. So you can clearly see the density difference between the internal whorls and the outer shell. And from this 3D model, we can look at a 2-dimensional cross-section, a 2-dimensional slice that shows the internal whorls as much as we would have looked at them if we were to actually physically cut or polish the specimen. And from this slice, 2-dimensional slice, I extracted the shape of 3 consecutive outlines of internal whorls. And the shape was captured using 30 evenly distributed semi-landmarks that were distributed from an equivalent anchor point. And this same procedure was conducted for the 2-dimensional images. So here we have a polished surface of a Middle Jurassic specimen. And the outline of 3 consecutive whorls was extracted and characterized again using these 30 evenly spaced semi-landmarks. This procedure was also used to extract specimens from mass accumulations of nerinids. Nerinids are known to form these massive fossil beds that can, and specimens can reach a density of hundreds of specimens per square meter. And these nerenid fossil-rich beds can extend for kilometers, and they're used as stratigraphic marker beds. So on the left here, we have a specimen sample of a mass accumulation of specimens from the Jurassic of Tanzania. And I manually segmented specimens in 3D and extracted them from the host rock. So here we have the 3D representation of the specimens in color and the host rock in gray. And for each individual specimen, I was able to select the 2-dimensional slice and characterize the internal whorl shape in a similar way. So from the coordinate data of the semi-landmarks, we can get a representation of the shape and test for variation in shape using multivariate analysis. So this is a discriminant analysis. Discriminant analysis is a form of PCA, and it shows the maximal variation between given groups. And it's again based on the multivariate data of the semi-landmark coordinates. So each dot here represents single longitudinal internal world section, and the different colors account for the 5 different families. And the confusion matrix below shows the number of cases that were correctly and incorrectly assigned to each of the groups based on the discriminant analysis. So overall, we have a good prediction of 73% correctly classified after jackknife resampling. And there's a good separation in the discriminant analysis of the blue and red families here. However, the 3 remaining families overlap in shape space. I'll talk about this in a minute. But if we group these 3 families together, we see that there's a significant difference in shape between these 3 different groups. So let's now zoom into the shape variation of each group using a PCA. Again, so each point here represents a single internal world section, and the lines here connect the internal worlds of the same specimen. So the first 2 PC axes, if we zoom into the blue group from the previous discriminant analysis graph, so the blue group represented here in the PCA, and we can see that the first 2 PC axes here account for 61% of the overall variance. And we can see that the shape of this group is the most complex, the internal shape. And we can see that the based on the shape analysis, taxa from so worlds from the same taxa group together, and we can see this as well for the red group from before, and again, specimen worlds from the same specimens from the same taxa group together. So we can say that this geometric morphometric method differentiates not only families for these 2 groups, but also even taxa to the taxa level. For the 3 overlapping families from the discriminant analysis before, here we have those in PCA, we can see that they overlap regardless of the data source or the sample size. And this suggests that there may be no true difference in the morphology of these families. And actually, if we look into the literature or the taxonomic history of these families, that this is supported by the fact that they're known for their complicated systematic history, which has been the focus of strong debate. So there's a good reason for us to think that they may not be actually separated. So to summarize this chapter, I developed a new quantitative method to identify Norenikin Gastropons, which are one of the most abundant and widespread groups of the Mesozoic. And the method is very robust and can be applied to a wide range of data sources, including museum collections, online databases, and placed and published papers. And it highlights the need to reevaluate the taxonomic identification in the fossil record. So I now showed how I developed this quantitative tool to identify Norenikin Gastropods, this key component of Mesozoic shallow marine environments. And our ultimate goal, if you recall, is to employ this method on the Norenikin assemblage to reconstruct the paleo environment. So I thought, let's take this method a step further from quantitative to fully automated using machine learning. And in the few remaining minutes I have, I'll describe how I developed an automated taxonomic identification tool for Norenids to test for their faunal response to environmental changes in the Jurassic. So in the Jurassic, there are 2 major biogeographic provinces that are recognized. The southern Tethyan realm, that's located around the tropical Tethys Seaway, and the northern boreal realm that's centered in Western Europe, and the Middle Jurassic sequence in Israel, which is located close to the Paleo equator, belonged to the Tethyan realm, and that extended from North Africa through the Levant and all the way south to Madagascar. And during the Jurassic, the opening of the North Atlantic and Middle Jurassic allowed Arctic currents, Arctic colder water currents to reach Western Europe. And as a result, the biogeographic distribution of shallow warm water fauna from Western Europe shifted southward. And this southward shift has been termed the boreal spread hypothesis. And it's been suggested for different fossil groups, including Norenids. However, its magnitude and extent were not quantitatively evaluated. So the goal is to use machine learning to test for the biogeographic shift of neurinids in response to environmental change. So we can break down this goal into several steps. And today, I'll talk just about these first two steps that I took to test for this variation with paleo latitude. First, I'll show how I expanded the database, and then I'll show how from this increased data set, I trained a neural network to classify images of neurinids. And I'm currently working on using this machine learning model to test for the boreal spread hypothesis, as I mentioned. So this is still in progress. So in recent years, there's been a tremendous increase in the digitization of specimens from museum collections and from the literature. However, the case study I showed of neurinids in the previous chapter really highlighted the need to update and validate the taxonomic data in these databases. So our image tagging problem, our image labeling problem still stands. Many taxa were described long ago and have remained poorly described, so identifications may be difficult to verify and may result in possible misinterpretation and inaccuracies. So we can't just download all this data and use it. We need to construct a larger database that has these tagged images to test our hypothesis. So the first challenge is, where can we acquire more images of Jurassic cross-sections of these neurinids? So if you recall, in the previous chapter, I used a bunch of different data sources, and I exhausted all of the available images of cross-sections of neurinids from the online databases and from the museum collections that I used. So to increase the data set, the remaining data source we have is images in these published papers, images of cross-sections in journals. So our first goal is to get a minimum of 100 images per category to train a model. So to do so, I compiled a list of references with potential images. And then I used freelance platforms such as mTorque and Upwork to engage multiple independent users to search for the data simultaneously. And out of the reference list of over 500 papers, about 200 were successfully recovered. I then manually filtered all these papers for the relevant images and extracted their metadata. So what's the age, formation, and locality or paleo latitude. And overall, I was able to increase the data set by over 170 new images, which is considered very big for a paleontological image-based data set. I then used supervised learning, which means training a computer to recognize patterns from labeled data. So the data set is divided to training, validation, and testing sets. And generally, what's advisable is an 80-10-10% ratio. So the images were assigned based to groups based on the results of the geometric morphometrics analysis from the previous chapter. And then I trained a model in Python using several packages, as you can see here. And these represent our three groups, predefined groups, based on the geometric morphometrics analysis. So very briefly discussing the results of the model, or how well can the algorithm assign images to their correct label. So here we can look at different definitions of model success. Overall, the accuracy of the model, or what fraction of the predictions our model got right, was very high at 89%. And looking deeper, we can compare the model's performance in the validation set for each label using this confusion matrix. So in an ideal model, all the values on the diagonal would be high and all the others will be low. So here we have for the three groups of neurinids and also a none of the above group, the model performs really well for this one group of the three combined families from the previous chapter. And it has minor confusions in labeling the other two groups, which is expected, considering that this group was the largest within the data set. And here we have examples of cases where the model correctly predicted the label for the image. So just to summarize this part of the talk, we set out to test this biogeographic shift of neurinids to track environmental changes in the Middle Jurassic. And to do so, I showed how I compiled a large data set of images of neurinids that I labeled based on the results of the geometric morphometrics analysis. I showed how I trained a model, classify the images based on our 3 predefined groups from the geometric morphometrics analysis. And after training, the model actually performs remarkably well with an accuracy of 89%. And I'm not working on using this machine learning algorithm to test the database for variation in paleolatitudes in the Jurassic, based on the images metadata, so the geological age and locality. And I want to conclude this talk and incorporate all the different, very different projects that we saw. So I used geoinformatics to reconstruct the Paleoenvironment of the Middle Jurassic. And I showed in this talk how I incorporated various tools, including multivariate analysis, micro-CT scanning, geometric morphometrics, and machine learning. And in each of these chapters, I showed how these methods can be used to reconstruct Mesozoic paleoenvironments and faunal response to climatic changes on a local, regional, and a global scale. I want to thank you all for listening. And I want to thank my supervisors, Yael and Folstenberg and Rivka Rabinovich, my advising committee, Uval Mogilabin and Amot Sagnon, all my collaborators, and to all the people who helped and gave very productive and encouraging feedback. And this research was funded by the Ministry of Energy and the Ministry of Science and Technology and the EU Synthesis Project. Thank you.
All right. Thank you so much for a great talk. Questions? If you have a question, please can either raise your hand or open your unmute yourself and ask. And hopefully you won't there won't be too many of you asking the same time.
Can I ask one?
Go for it.
Do you hear me?
Yes.
Yes.
Very impressive, Yael. I was wondering about the validation of the morphometry while you were using these 3D sections that you synthetically recovered from the 2D section from the 3D data. So do you have validation for this?
So that's actually a really good question.
Works.
Yeah, so let me go back to that slide. And yeah, so validation was actually a very important part of the process. So, okay, going back to this slide. So I think what you're asking is, you have this 3D model reconstruction and then you switch to this 2-dimensional view. How do you know that you chose the right 2-dimensional slice?
Also,
yeah, right?
even
right.
this is part of the question, but not all.
So
let me make it clear that I also want to know that the 3D structure that you have is accurate enough. I mean that your basic data is really representing.
Well, I have so the scanning data set was pretty big for paleontological studies. Most studies scan only 1 or 2 specimens, and I was able to extract the 3D morphology of over 30 specimens. So the data set for these 3D analysis was pretty large, but I wanted to increase the database to get a more statistically significant identification of the structure. So I had to reduce one variable. I had to reduce one dimension to do so because I wanted to incorporate more images. And you know, there's always a trade-off of the number of variables you look at and your sample size. So reducing it from 3D to 2D was the optimal choice. And I actually performed a lot of validation tests for this process. So this was published this was detailed very extensively in the paper because this was a primary focus of the method, making sure that we don't miss out on any of the internal structure when moving from 3D to 2D representation. And so I tested this in a couple of statistical ways, both both on the number of segments you need to do, how many times do you need to capture the internal shape of each internal whorl, and also what's the difference between different slices that are close together in the 3D representation. So what CT stack did you choose? So those are two different types of statistical analysis, and they both showed that the actual location of the CT stack didn't really matter, and also that because we use so many semi-landmarks for each internal structure, the number of times I extracted the shape just basically remained the same, gave the same indication to shape. I hope that answered your question. Oh yeah, I think Uli, I think you're on mute.
Sorry, I was muted. Ali, go ahead. Sorry.
I have a question here. Can you go back to any of the Goldberg's cross-sections? Any of them? It doesn't matter which
Sure.
one. Yeah, that's fine.
Okay.
So following this up section and your results, you indicate a trend of shallowing when you look at a temporal scale. But in fact, what we see when we look at the entire cross-section is alternations between a sea that supports reefs and a sea that doesn't support reefs. Because overall, the white patches in their volume or area are much larger than the ones that are colored. So we have hundreds of thousands or maybe millions of years of an ocean that doesn't support any reef, regardless of what kind of reef. Can you tell us something about those periods? Why do the reefs disappear completely and then return?
Yeah, actually, that's a really good question. So the southern reef.
It wasn't prearranged.
It wasn't. So the location of the Makhtesh Gadol is within this larger band of the southern reef belt of the Tethys. And this southern reef belt is more impoverished in reefs and actual reef structures relative to the northern reef belt at the same time. So the notion today is that temperature, as I mentioned, specifically for Makhtesh Gadol, but also temperature in general for the area, was this limiting factor that restricted the success of corals and sponges in the area. So these patchy reefs that we have in Makhtesh Gadol are actually the exception, are the species that manage to survive to build these structures despite of the unfavorable conditions. And within this sequence, we have two horizons that show relative more success. But if we compare all of this data to northern to reefs in the northern reef belt in the Jura Mountains, they show much lower species diversity. They don't form bioherms. They're much less successful globally.
I have a question, if I may.
Go ahead.
Hi. You took a field, a scientific field, which we all were used to look at it as the descriptive field. So that's not how we learn paleontology, and you quantified it. So I have a question. Maybe you said that and I didn't get it, but I want to clarify it. What when you quantify this field, how much of your observations are really new? And how much is just to just see better things that previously were described already? So let's say we take Goldberg and Hirsch, all these guys, the older generation.
Yeah, they worked on the sequence, yeah.
So, and we ask them what is really new and what is just, you know, improvement of the things that they saw descriptively or qualitatively, excuse me.
So let me go back to this slide, and I think that will answer your question. So you ask, what's new here, right? We know that the Jurassic is this shallow marine platform. What's new? But we don't know the details. We don't know the details of any of the for any of the Middle Jurassic sequences. Based on the single occurrence of a fossil, you can't really reconstruct the depositional environment. So what I do is I don't only look at one taxonomic group or one species of fossil. I look at the entire fossil assemblage, and using these methodologies that are also used in marine ecology for modern reefs, we can get a statistical understanding of the changes in the community structure and composition of the fossil assemblage, and then get this quantitative assessment of the environmental conditions. So you say it was shallow and hot. How shallow? How hot? I think that's what the addition that you're looking for in my data. True, past researchers have said some of these species models for the Jurassic, but they didn't have any quantitative data to support their claims, and they didn't have the details, the high resolution information.
Yael, can you specify for Zohar exactly what new shift you saw that Hirsch and Goldberg didn't see in the Mahdi, for example?
I'm sorry, Yael, you were breaking up.
So if you could specify what is your own work, your own quantitative work, what did that expand on what Hirsch and Goldberg knew in the funnel? What you're talking about, the shift, the main shift that you saw is the first time that this was ever discussed.
Right, so as Yael kindly reminded me.
Maybe you want to just give that again.
So to give, to go back to this temporal reconstruction, both from Hirsch and Goldberg's information, both on the lithology and some general identifications of the fossil fauna, we couldn't reconstruct this variation, this high resolution variation in the environmental conditions in the structure, in the sequence, right? So this is directly from my own quantitative work on the fossil assemblage. We can get a better sense of the shallowing and deepening of the water curve, of the and the variation in turbidity and nutrient levels within the sequence. So this is high resolution detail that we didn't have before.
Okay, can I ask some questions?
Go ahead, Chaim.
Yeah. Okay. Just when most of us talk about environmental conditions, the first thing in geology, the first thing we look at is the sediments. We can do things with the sediments, but first look at the sediments. And if you look, for instance, at the Saudi Arabian section, there's a very prominent column over there with the simplest, most elementary sedimentological data about mudstones, wacky stones, pack stones, and grain stones, which is right there next to the rest of the data. And it's very prominent. It's very easy to make a sedimentological inference for a particular part of the section. One very simple example, for instance, is the oolites versus perhaps frame stones or something like that for the coral reef parts. Those are there in the Saudi Arabian material, but I don't really see that in the analysis of either the Makhtesh or perhaps the Hermon sections. But I'm sure the information is available. These are sections that have been studied a great deal. And there are some very significant environmental correlations connected to the sedimentology long before you get to actually interpreting the form of the, okay, yeah, Yael, you're right.
I'm with you, I'm with you. Go ahead.
Yeah. So I would like to see that column, for instance, in the picture that we're seeing right now. I'd like to see a column like that, and I would add one more thing, which I'm sure exists, and that is that we know that southern Israel, and for that matter, the difference between the two sections in the Saudi Arabian material that you showed, are the Negev sections are more proximal to a clastic source, a silicic clastic source, as is the for the western section of the Saudi Arabian section you showed, as opposed to the more carbonate rich that the eastern Saudi Arabian section would be, and also presumably different parts of the section that we're looking at over here. The part that's marked as Kidod, or so-called Kidod, we know we have some difference of opinion as to the nomenclature here, but we'll put that aside. But there presumably is a high, fine siliciclastic component in that middle part of the section, which would have a lot of bearing on, for instance, whether we get reefs or not. I would have liked to see this basic. I don't think that it's just simply from my knowledge of the material. I don't think it's necessary to do a new study in order to put this database in there. And it seems to me that it would provide a lot of rather significant environmental information and perhaps an explanation for many of the changes that you see, especially, for instance, in the changes in the uppermost part of the section, where you have alternation between several different environments interpreted as mesotrophic or oligotrophic, and clearly the sedimentary pattern should have some bearing on this part of it. That's my first comment.
But hold on, Hein. Maybe it was a long comment, so yeah. Okay. Maybe we can let Yael answer that, and then
okay.
We'll move on.
Yeah, so I just want to add, Chaim, that I agree that the sedimentology of the sequence plays an important role here, and it's actually incorporated within the analysis. As I mentioned, I followed Musi Goldberg's subunits in very high resolution throughout the sequence. So we have the sedimentological data for each of the patchy reef horizons, and that's definitely incorporated within the analysis.
Okay, we maybe can have a private conversation about this. There is one other interesting aspect of this. This is basically what my question is, and that is that there's another paleontological database that is presumably available. Where it is at the moment, not so clear, but I'm sure it's there. And that is that together with the macrofaunal record, there's also a microfaunal record. Certainly of foraminifera, perhaps of other species and other groups. There's also information about calcareous algae, and these are also paleoecologically significant groups that I think would have bearing on what kind of environment we have over here and what groups would correlate with different kinds of things that would be nicely included in the diagram you just switched to over here. Yeah. Okay. And I have a third question, but maybe you want to say something here.
Yeah, so I just want to mention that there's a lot of other information on taxonomic groups, and we have here a limited time, so I just showed some of the data I have for the mollusks from the same part of the sequence, and that's also quantitative data. But I agree, there's a lot more information to incorporate here that could shed more light on this sequence.
Okay, and my third question is about the second part of your talk, and that is the analysis of the Norenikin Gastropod matrix, basically. Actually, this is
internal structure, I would say, not matrix, but yeah.
I'm very excited about this because we can see that we have Norenikin groups over here in what is interpreted as very shallow environments. But we have in the deepest environment, according to your analysis, we have a much more significant Norenikin component.
True.
And of course, a very good question would be if we carried down slope along this trajectory, where would the Norenikids be? Because in your opening about this part of the analysis is that we all know that Norenikas are very prominent members of the very shallow warm water assemblage, and this part of the diagram shows that maybe the distribution of this group is significantly more complex than that very basic statement. But there is one other aspect of the numerical analysis that we saw, and that wasn't mentioned, and that is that if one were to make a third axis, which included all three groups of Norenikids, that would put basically to the unmathematically trained eye, the first thing that I saw in this analysis was that these three groups are very different in terms of what would just be called complexity, maybe even just the number of folds. You have one group which has a very small number of folds, a third group which has a large number of groups, and the central group which, or most of the Norenoids, well, three of the Norenoid so-called classes fall in the middle over here. And of course, the question that if you were to put these along a third axis, you have a first axis and a second axis here, but you could put a diagonal axis over here in which they would fall along the complexity axis. And the first question we're asking, is this axis an environmental thing? Is it an evolutionary thing? These are large questions, which obviously would be the target of the end result of perhaps a lifetime analysis of this group. But this is from paleontological, perhaps environmental point of view, one of the first things that one would ask about it. So this is more just a simple question about how another way of using this analysis, and is this part of it telling us something? Obviously, your analysis is in much too early a stage in order to be able to come to a reasonable conclusion about these things.
Yeah, so I want to be very careful with how much systematic importance I attribute to this method, but I think it does highlight the fact that these three groups, which to the visually they seem very, very similar to each other, may not be taxonomically different. And to be honest, I looked at several different axes of the variance in shape space, looking at the third diagonal axis that you mentioned, and it didn't actually have any bearing on the separation between the predefined groups, despite performing a discriminant analysis. So I was pretty confident in my decision of grouping clumping these three families together, and this was supported by the fact that they were very under debate in the taxonomic literature. Even the most up-to-date revision of these groups remains in some, has some unresolved questions about their taxonomy.
All right. I think that we're way past time.
Yeah.
So, well done. If you have more questions, you can. I'm sure you can get in touch with Yael and ask later. All right. So I'd like to thank our speaker again, and thank everybody that joined this session. And at least for the Hebrew University, we have another seminar coming this Wednesday. All right, so I'm going to send out an email about it later. All right, going to adjourn this meeting.
Thank you.
Thank you, Yael.
Thank you very much, Yael.
Thank you. Bye.
Hmm.